Now we will see the digestive glands. Digestive glands. Digestive glands. So this is all about, already in the previous class we discussed about alimentary canal, the entire digestive system which is started from mouth to ended with anus. So in that we discussed all the different parts. Now we are going to discuss about the accessory glands. Accessory glands means they are associated. Okay, some glands they supports from outside, some glands which are present within the wall of alimentary canal. So here first of all we will discuss about the accessory glands which are present outside. Later we will discuss about the glands which is present in the walls of the gut. Okay, you see there is a picture you are observing, the gland which is associated with okay, the our particular alimentary canal here in the buccal cavity. What are going to open in this buccal cavity we will see now. Okay, so right, you see this part here, you are observing about salivary glands, the glands which are going to get association, that are salivary glands. Salivary glands. Okay, right, you know saliva, when you see a tasty food we will get salivation. So like that generally we will feel. Okay, if you saw whatever the food you like, you will get a neural sensation. Then immediately we will see the salivation in our mouth will become wet. And another important thing is there, the saliva is also useful for okay, digestion of carbohydrates and slightly lipids also digested. So the saliva which is going to get through the salivary glands, where these salivary glands are located we will see now. Generally, you know, so below the ear pinna, suppose here you can see the ear. Okay, see this is the ear. So see this here in this picture you are observing near to the ear pinna. You see now? Yes, this is the ear pinna. So near to the ear pinna inside, okay, you can see these are the glands called parotid glands. Parotid glands. Okay, what are these glands? These glands are called parotid glands. The most important glands in the salivary glands. Among the three types, in actually human being there are three types of salivary glands are there. But in other animals, especially in the mammals, you can see 4 pairs of varieties of salivary glands are there. But in human, there is only 3 varieties of salivary glands are present. Right. So now we are moving to the first one. What is that? Parotid gland. So where is its location? Below the ear pinna. Okay. From below the ear pinna, it has a duct, a special duct. This is called Stenson's duct. What is this? Stenson's duct. This Stenson's duct, through the Stenson's duct, the saliva released into the buccal cavity. So like that in this picture also you can see, so very clearly you are observing this duct, Stenson's duct. With that it will open here in this area, here it will open in this area. So like that. Okay, now let us move to the second one. You see in this second one you can see here submandibular, we can also call submaxillary, submandibular, submandibular or submaxillary. This gland, why we said like that? Because it is present where the upper jaw and lower jaw is going to mix. So in this area, so that's why it can named as submandibular, submaxillary. So from these there is a gland called Wharton's duct. What is this duct? Wharton's duct. Wharton's duct. So these Wharton's ducts releases saliva into the buccal cavity. So here in this picture also you can see, so very clearly you are observing, see this one. You see everyone? So this part. So here you are observing Stenson's duct and here you are observing Wharton's duct. It is very important in the competitive exams. They will ask you about the ducts. Okay, the next one, another one is below the tongue. You see, I am going to give you in red color. This is all about, okay, you see this is the tongue. Okay, so this tongue. So below the tongue also you can see the salivary gland. As everyone knows this, because we will see, okay, which is present, if you fold your tongue below, you can observe this is all about sublingual. Sub means down. Just now you see submandibular, submaxillary, because which is present in the downside. You see here it is in the downside. So here also you see this is the tongue. Below the tongue you are observing sublingua. Lingua means tongue. So anyway here we are writing this is about sublingual. Sublingual. So like that there is 3 pairs of salivary glands are present in human body. One is parotid, another one is submandibular or submaxillary, and the another one is sublingual. So like that there is 3 pairs of glands located in this particular area. Just now I told about the one more gland, that is infraorbital, present below the eyeball, but it is absent in the human beings. In animals we can see, okay, below the, okay, suppose this is a eye orbit. So below this you can see this is infraorbital. Infraorbital. But this infraorbital is absent in man. This is also one of the important point in competitive exams. Okay, absent in man. In other animals you can see there is 4 pairs. Okay, saliva. Saliva is a mixture of amylase. Amylase enzyme is also called ptyalin. Ptyalin. Salivary amylase is also called ptyalin. Alpha amylase. Somebody they will also use this type of word. Alpha amylase. Salivary amylase. Ptyalin. So like that the words we can use. And mucin. Mucin is also present. Why? Because mucus, which is useful for holding the food together at the time of chewing. When food is manipulated while chewing, at the time mucin is very important to hold the food particles together to make bolus. Bolus means a ball of food. And the next one is amylase and lingual lipase. Lingual lipase. Okay, this is very little. Okay, which one is very little amount and lingual lipase is very little amount is this. But the most important enzyme is ptyalin. Ptyalin is going to work on the carbohydrates. Carbohydrates are converted into disaccharides. Nearly 30% of the starch. 30% of the starch is going to get digested into disaccharides with the help of this. We will discuss this part clearly in the process of digestion. But anyway, amylase enzyme is very important. So already in the mouth itself, the chemical digestion is started with the help of enzyme. What is that? Carbohydrate digestion. Okay, so this is all about with the help of chlorine ions, also very important for this process. What is the next part? Here, esophagus. Can you see any digestive glands here in this area? No. In the esophagus there is no digestive glands because the food is not going to store here. Once we engulf the food material, that is directly passing into stomach. So that's why there is no need. Esophagus is fully lined with mucus. So that's why food directly coming to the stomach. So that's why esophagus region do not consist any enzymes and glands. Next, coming to the part, this is all about, you know already, what is this part? Stomach. Okay, stomach. Okay, right. So in the stomach, what are the glands are present? They are called gastric glands. They are called gastric glands. Next part, what is this? Small intestine. So one by one we will go. So first we will discuss about gastric glands, which are present in the stomach wall. These are not associated. This is already present in the wall. But salivary glands are associated glands. They are present outside. They are giving support from outside. But now here it is not like that. Here there is a direct release of gastric glands. Now we will see the gastric glands. Yes, you see. So in the stomach you can see many rugae are there. You know these foldings you are observing here, these are all stomach foldings. This is all about the stomach wall. Already you know stomach wall consists serosa, longitudinal muscle, circular muscle, here and there oblique muscles are there. In previous class we already discussed about this point. So, and mucosa, right? So in mucosa what you are observing? Lymphatic vessels and as well as artery and as well as veins. But the important thing is you see the mucosa, whatever the foldings you are called, okay, that is all about the mucosal foldings. These are the small foldings called rugae. These are called rugae. Yes, if you zoom this part, particularly this area, yes, you are observing here. Okay, so these rugae consist, the base of the rugae, there are many varieties of glands are there, cells are there. Okay, so what are that cells we will see now. Generally in this area you can see this is cardiac part, okay, this is the fundic part and this is the pyloric part. Cardiac region, fundic region, pyloric region. Okay, so directly when the food comes, directly it will felt into the fundic region and also into the pyloric region. So this region above the fundic region, you can see this part is, okay, it's filled with gas. Cardiac region is very near. Okay, so that's why this part cardiac sphincter. Cardiac sphincter. This already you know about this cardiac sphincter. Cardiac part. Cardiac part is specially meant for releasing mucus. What is secreted from the cardiac part? Mucus is secreted. Okay, so another thing is in the fundic part you can see the different types of cells you can observe. In the fundic region there are different types of cells are there. One is fundic cells, auxentic region also you can say auxentic cells. Auxentic cells. We can also call parietal cells. Auxentic cells or parietal cells. Auxentic cells and parietal cells which secretes HCl and Cassell's intrinsic factor. These are very important. HCl and Cassell's intrinsic factor are secreted from auxentic cells or else called parietal cells. You know the importance of HCl? Okay, so here I am writing HCl. It is a acid. So that's why if any bacteria are there, these bacteria in the food are going to be killed by the HCl. That is one of the important use. And it can convert pepsinogen to pepsin. Another important use is there about the HCl. Prorenin. Prorenin is converted into renin. So like that, here inactive enzymes, pepsinogen, inactive enzyme, prorenin are converted into active enzymes like pepsin, active enzymes like renin. That is the importance. So HCl is enzyme activator and also HCl is useful for killing the pathogens. How much pH it is? The pH is almost, the HCl pH is almost 0.9 to 1.2. Sometimes, okay, it may be 1.5 also. So, but anyway, this is the number here, 0.9, approximately here and there slightly to the 1. So that much acidic HCl is present in our stomach. So that's why mucus secretion is very important. Once again you can see here, suppose you see this is our stomach, here the HCl acid is there, and the important thing is this is very thick mucus lining is there. What is that? Mucus. Why? Because this mucus lining protects the excoriation. What is the meaning of excoriation? Damage caused by acid. That is prevented by the mucus. So that's why the acid is there, but there is no effect to the stomach. Otherwise we can see the ulcerations because of that acid. That is called acidity. Generally we will use in general terms. Right, anyway. So like that HCl, that is the importance of the HCl. And another Casel's intrinsic factor is there. Casel's intrinsic factor. Casel's intrinsic factor is useful for vitamin B12, absorption of vitamin B12, absorption of vitamin B12. This is also called cobalamin, cyanocobalamin. That is important for absorption of vitamin B12 from the food. So like that, the parietal cells, you are observing the parietal cells here. You see in this photo, these parietal cells are useful for that process. We can also call that are auxentic cells. Another type of cells, these cells are called chief cells. What are that? Chief cells. We can also use another word, peptic cells. Chief cells also called peptic cells. Yes, first we discussed about parietal cells, next we discussed about chief cells. These chief cells will release pepsinogen. Pepsinogen. Another one is prorenin. You have to write double N. It is important. RENNIN. That is very important. Why? Because another renin is there. You know that renin is helpful in the kidney, RAS. So here you have to write renin, double N. But anyway, pepsinogen, prorenin. Can you say that these are the directly they can work? No, they cannot directly work. Why? Because pepsinogen and prorenin both are inactive. Both are inactive. What happens if directly the enzymes released in the active form? Generally, if the enzymes are released directly in the active form, they start digesting the wall of alimentary canal. Suppose you are hungry. Suppose, okay, so you are not taken food for one day or one part of the day. Automatically, these enzymes, when it is released directly, they will act in the alimentary canal. So that's why our alimentary canal starts getting digestion. So that's why all the enzymes are proenzymes, means inactive enzymes. When we will eat food, then because of some activators, they are converted into active form. Otherwise, okay, there will be a trouble that is related to autodigestion. That process is called autodigestion. So that is prevented. So that's why they are released in the inactive form called proenzymes. Okay, so these are the two main things, and another one is there, mucus. This is called neck cells. Another cells are there, neck cells. Neck cells. See, all here is very clear in this diagram. You can see mucus cells, neck cells, parietal cells, chief cells. Everything you are observing clearly in that. Okay. Now, neck cells, what they will do? They are also useful in the secretion of mucus. So, here the mucus secretion is there, cardiac region. Even in the fundic region also you can see the mucus secretions. And the next, pyloric part. These pyloric part consists the special cells called G cells, and also they consist mucus secreting cells. Mucus secreting cells. Okay, mucus secreting cells. G cells. What about the G cells? See here. Here in this, the bottom yellow color you are observing, this is all. The bottom layer. That is G cell. G cell is specially meant for the releasing of gastrin hormone. What is this? Gastrin hormone. Not enzyme, hormone. And endocrine system is also involving, which is going to stimulate the secretion or inhibit the secretion. So here gastrin hormone is very important to activate all this, whatever we discussed this, all cells are specially activated by G cells. G cells releases gastrin. This is a hormone. What is the use of gastrin? Gastrin stimulates gastric glands. Gastrin stimulates gastric glands. Yes, we completed our part and we are moving to this particular region, duodenum. Okay. Now, you know this is the pyloric sphincter. After coming out from that region, we can see the C-shaped duodenum. C-shaped duodenum. Yes. Then next, here is the liver. Okay, generally bilobed liver is present in our body. Yes, this is all about the liver. Okay, and another gland is there. What is this? Pancreas. Pancreas. What about this? Liver. So, here in the duodenum, there is a union of these two particular, you see, there is a bag. Okay, so this bag and its connection. Okay, so like that you are observing this particular part. Here you can see, what is this? Gallbladder. 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 So liver, and as well as here you can see from pancreas also there is a connection. This connection together they will open. So from here bile juice is coming, from here pancreatic juice is coming, and finally they are opening into OD sphincter. Through OD, what is this? Sphincter. Through OD sphincter it is going to get open. So this immediate part is called ampulla of Vater, and this part is called hepatopancreatic duct. You know this is pancreas, and from here the pancreatic duct is coming. From here you can see this is gallbladder. From here common bile duct is coming. Common bile duct and pancreatic duct. This is common bile duct. Common bile duct, and this is pancreatic duct. Pancreatic duct. So pancreatic duct, common bile duct, together hepatopancreatic duct. Starting it is with ampulla of Vater, and that ends with OD sphincter. So these are the parts which are going to discuss now about the liver, the largest gland in our body. Then we will move to pancreas. Later we will discuss about the small intestine, intestinal glands. So what are left out? Liver, pancreas and succus entericus means intestinal juice. Right. Now bile juice, pancreatic juice, intestinal juice. Like that we will discuss about the three different types of enzymes. Okay. Now first we will move to the liver. Now our topic is all about liver, right? Now you see in this diagram, this is all about stomach. Yes, you see, what is this? Gallbladder. From gallbladder what is coming? Cystic duct. From the liver what are coming? All together common hepatic duct, that is opening with, you see, common bile duct. It is given common bile duct, but actually it will mix it with pancreas. Here in this diagram you can see a direct path, but actually it is connected with, so actually here there is a connection. Right. What is this connection? This is all about coming through this. This is all about pancreas. So this is the way. So now according to this it will be OD sphincter. OD. Automatically it will be water. Ampulla of water. This is duodenum. So this is the way how the passage is there. Now we will see one by one clearly about liver. Okay. Liver. Yes, liver is a bilobed structure in a human. It is located in the right side. Left side what is present? Stomach is there. Right side below the diaphragm there is a liver. With how many lobes? 2 lobes. You see here one, here one. There is 2 lobes. It's one and one. Okay. So from this there are many ducts, many connections are there, many ducts, many branches. So like that all together they are opening and they are connected to gallbladder, and finally they are opening into duodenum. Okay, so what are these we will see. What is this and what is this and what about this? Yes, this is common hepatic duct. Common hepatic duct. This is, what about this particular? Cystic duct. Cystic duct. So common hepatic duct, you see this, common hepatic duct, cystic duct together forms common bile duct. What is this? Common bile duct. So remaining you know very well. What is this? Ampulla of water, OD sphincter, everything. Right. What about this? Gallbladder. Gallbladder. Right. Now we are moving into the internal structure of liver. So from where this bile is coming, from where this is collected, we will see now. Now the internal structure of liver. Suppose if you cut a piece of liver, if you see this, a piece, is this a small piece? As you are observing, this is a small piece. If you zoom it, you can see this particular part. Right here it is divided into many hepatic lobules. You see these are the structures. This is one hepatic lobule and this is one hepatic lobule and this is one hepatic lobule. So like that A, B, C, D. So like that many lobules are there. What is present? What is present in these lobules? Yes, lobules are called structural and functional units of liver. What are called structural and functional units of liver? Hepatic lobules. From this you can take a small part, a small piece, and under the microscope if you observe you will get this. Right. Inside there is a common vein. From that, this is called central canal or central vein. We can also use the word, that is very important, central vein. And you see the radiating. What about this? These are the cells. How the cells are arranging? Cells are arranged in the cords like. So the cells are arranged in the form of radiating manner. So this is the way. So like that many rows. So what is this row? This row is called hepatic cord. What is that? Hepatic cord. What are these circles like structures? These are all hepatocytes. What are these? Hepatocytes. Right. So this is hepatocyte. So hepatic cord, hepatocyte. What is the space between two cords? This is called liver sinusoids. Sinusoids. What are liver sinusoids? The gap between the hepatic cords. This is one hepatic cord, this is one hepatic cord. Between the space is called liver sinusoid. What about this part? Just now I told. What is this? Central vein. Central vein. Okay. And one more thing. You can see there is a small border, outer boundary you are observing. This is all outer boundaries. This outer boundary is named as Glisson's capsule. What is this? Glisson's capsule. This Glisson's capsule is very thin outer connective tissue which is going to get the separate all these hepatic lobules. Once again I am repeating, the hepatic lobules are the structural functional unit of liver. Okay, central vein, hepatic cords. In each hepatic cord, hepatocytes are arranged in the form of rows, and the gap called liver sinusoids, Glisson's capsules. So this is, so like that all, okay, remaining all these parts also the same. Understood? So these cells, hepatocytes releases bile. Hepatocytes releases bile juice. These bile juice will pass. So now you see hepatocytes releases bile juice. These bile juice through hepatic ductules opens into hepatocytes, bile juice, hepatic ductules, from there common hepatic duct. Okay, you can also have hepatic duct. See, two hepatic ducts are united to form common hepatic duct. Common hepatic duct to come to the cystic, and here it is stored. Gallbladder is for storing the bile juice, but not for the secretion of bile. So bile is secreted from the liver cells and stored in the gallbladder. That is one of the important point. Anyway, so this like this is the passage. If sometimes they will ask you how bile is passing, how it is opening. So you can say like that, hepatocytes releases bile juice will enter into the hepatic ductules will form hepatic duct. All hepatic ducts are together forms common hepatic duct, and it enters into the gallbladder and comes out through cystic duct, common hepatic duct, and finally hepatopancreatic duct. This is hepatopancreatic duct. Through OD sphincter it opens into the duodenum. You see, this is the OD sphincter. So like that it opens into the duodenum. Okay, this is all about liver, the one of the important okay structure in our body. So what are the functions of liver we will see. First we discussed about, okay, it secretes, and liver secretes bile juice which is stored in gallbladder. These bile juice, there is no enzymes. This is one more point. Bile juice is coming from liver, but there is no enzymes. Cholate, tarocolate, like sodium potassium salts are present. Bilirubin, biliviridin, like that pigments are also present, but there is no enzymes. So it is not useful for the digestion, but prepares fats for digestion. How it prepares fats for digestion? That process is called emulsification. That process is called emulsification of fats. Make the fats into small particles, and also they forms coating. So then once lipids in our body, once the fats in our body, compulsory it has to be coated with bile juice. Then only lipase enzyme will work. Directly the fats, they do not directly acts on, okay, directly lipase enzyme do not directly acts on fats. Lipase enzyme compulsory works emulsified fats, works on emulsified fats. Anyway, so this is all about bile juice. Okay, another important use is there. Liver useful for carbohydrate metabolism. Carbohydrate metabolism. So here in the carbohydrate metabolism, what it is going to do? Carbohydrate metabolism lo first one is glycogenesis. Okay, glycogenesis. So means what? Whatever the glucoses are there, these all the glucoses together converted into glycogen. So what is the reaction? All the glucose molecules are converted into glycogen and stored. So that's why it is our energy bank, liver. So whenever you want glucose, glycogen again converted into reverse process. So like that glycogenolysis means again this glucose is converted into, glycogen is converted into glucose. So initially glucose is converted into glycogen, later glycogen is converted into glucose. This process is called glycogenolysis. So like that, the important thing is there, and sometimes gluconeogenesis is also there. According to gluconeogenesis, other than carbohydrates, means fatty acids, and also from amino acids, and also from pyruvic acid, glucose is prepared. Normally from where glucose will come? Normally glucose will come from carbohydrates. Carbohydrates end product is glucose. But here you see from fats, and it related to, that is other than carbohydrate. Amino acids related to proteins. Pyruvic acid. So like that, these are the different types of products. From that also glucose is synthesized. So that's why it's very important process. So like that, glycogenesis, glucogenolysis, gluconeogenesis. So like that, it involves in the carbohydrate metabolism. Function number 3, detoxification organ. What is this? Detoxification. organ. Liver is detoxification organ. Means what? See, this is all our alimentary canal. Simply I am writing GUT, gut. So here is the liver connection. So liver through hepatic portal vein. What is this? Hepatic portal vein. Through hepatic portal vein, it will receive all the materials from alimentary canal. So whatever the food we will eat, initially which organ it is going to be? They are going to be enter into liver. They are going to enter into liver through what? Hepatic portal vein. What is this? Hepatic portal vein. Through that it will enters into liver. Suppose if any poisonous substance is there, if any poison substances are there in our food, or we through water, or in the form of liquid or in the form of solid, any toxins are there, or alcohol is there. So these all the substances, where they are going? Directly they are going to liver. So liver will try to reduce the toxicity. This is the important. Reduce the toxicity. That's why generally, if any person consumes a lot of alcohol, chronic alcoholism, at that time liver is going to get damaged. So like that, the liver is very important, which reduces the toxicity before reaching general circulation. See, so that's why it is the first guardian angel of our body. That's why this is the guardian angel of our body. Ammonia is a very toxic product. Ammonia is converted into urea. Ammonia converted into urea. Right? So this process is called ornithine cycle. Ornithine cycle. This is one of the important, very star point. Ammonia is highly toxic substance which is released through our metabolic activities, protein metabolism. Through that, ammonia is formed in our body. That ammonia is converted into urea. So this is very important. Urea is not that much toxic. So that's why ornithine cycle, that is one of the important point. So like that, it can also lipid metabolism. Lipid metabolism stores fats, excess fats stored in liver in the form of triglycerides. In the form of triglycerides, lipid stores fats. And another important, produces heat, provides heat to our body, Why? Because it is glycogen is there. Whenever we need energy, whenever we need heat, so that glycogen is converted into glucose, then from the glucose energy released and heat is also released. That is one of the important thing which helps in the thermoregulation of our body, produces heat and produces or synthesizes, produces blood clotting factors. What is this? Blood clotting factors. For example, blood clotting factors: fibrinogen, prothrombin, etc. So they are secreted. And also anticoagulant, heparin, anticoagulant. Anticoagulant means which prevents blood clot in blood vessels. That is anticoagulant, heparin. So it secretes both blood clotting agents and also anticoagulant agents. So that is also one of the important use of liver. So like that, many uses are there related to liver. Cori cycle is also one of the important. You know, when our muscle gets tied, you know, lactic acid is formed. Okay, this lactic acid will reach the liver from the muscle, which will reach to liver. Then lactic acid is converted into pyruvic acid. Pyruvic acid is converted into glucose. Just now I told about neogenesis. What is this? Gluconeogenesis. Gluconeogenesis. So like that, from lactic acid pyruvic acid will form, from pyruvic acid glucose will come. This is called Cori cycle. Okay, so like that, another use is also there related to liver. Okay, coming to the next part, another part of these glands, just move towards the duodenum and also pancreas. First we will discuss about pancreas. Pancreas. Okay, you see here in the corner below the stomach, pancreas is present. Pancreas is called mixed gland. Yes, this is pancreas. Okay, here you are observing the pancreas. So it is present below the stomach and also in the corner of the duodenum. Flat gland. Okay, it is pancreas, which is also named as very famous. It is called mixed gland. The famous name of this pancreas is mixed gland. Why? Because it will do exocrine secretion and also it will do endocrine secretion. Exocrine and endocrine secretions. Exocrine, endocrine. What is exocrine and what is endocrine? We will see now. If you see the pancreas, under the microscope, if you observe the pancreas, you can see two varieties of cells. One type of cells called acinar cells. One type of cells called acinar cells. These are the cells. So like this, a group of cells. Acinar cells. Yes, acinar cells. And very few here and there. Islets of Langerhans. Islets of Langerhans. Acinar cells. So which cells are more? Which cells are maximum? Yes, acinar cells are maximum. Very few cells, islets of Langerhans. Islets of Langerhans will do endocrine function. Means islets of Langerhans will release hormones. But our part is digestion. Means related to digestion, acinar cells releases enzymes. This is the story. That's why pancreas is called mixed gland. Okay, so it releases hormones, it releases enzymes. So that's why from acinar cells it releases enzymes, from islets of Langerhans it releases hormones. Anybody can say about the hormones? In your previous classes, in 9th and 10th you may study insulin and glucagon. What are the hormones secreted by the pancreas? Means insulin and glucagon. But now our part, this is all about the digestive system, no? So we will discuss about the enzymes, about this, okay, particularly acinar cells which are going to give. This is trypsinogen. Trypsinogen. Number 2, chemotrypsinogen. Chemotrypsinogen, carboxypeptidase. Okay, and nuclease. Okay, and amylase. amylase and lipase. So here this lipase is called steopsin. Very, very important point. Steopsin. What is steopsin? Pancreatic lipase. Pancreatic lipase is called steopsin. Trypsinogen, chemotrypsinogen, carboxypeptidase, nuclease, amylase, lipase. So like that, the most important enzymes are secreted from the pancreas. These are very important, which will take first role to digest the complex food materials. Later, intestinal juice will work. But initially, which hormone is going to work first? Pancreatic juice. Because pancreatic juice is called complete digestive gland. Pancreas is called complete digestive gland, and the most important thing is the first action is always about pancreas. Then remaining action is about intestinal. We completed about liver, that is all about bile juice, and also we discussed about stomach and pancreas. Now we are moving to the gland. Can you say pancreas is accessory gland or within the it is present? No, it is accessory gland. Why? Because it is present outside, associated. Salivary glands associated. Liver one side, associated. Pancreas is associated. So like that, three main associated glands are there. Gastric glands and intestinal glands are present within the wall of stomach and intestine. Okay, so intestinal glands. Intestinal glands releases succus entericus. Succus entericus. Intestinal glands releases succus entericus. Okay, so here the intestinal glands we will see now. Succus entericus. So here, you know, if you observe the intestinal wall, in the intestinal wall itself, you can see the villi. So that is the duodenum and jejunum, and another one is ileum. Three parts are there. Okay. What is the specialty of duodenum? In duodenum, submucosa. From where succus entericus will come? From duodenum, it will come from submucosa. Okay. Next. And again, from submucosa, what are that? Brunner's glands. What are these? Brunner's glands are present. Brunner's glands. And from jejunum and ileum, from these two parts, mucosa. At the base of mucosa, what are that? Crypts of Lieberkühn. Crypts of Lieberkühn are present. So both will release this. Understood? Succus entericus. The succus entericus is going to come from duodenum, jejunum and ileum. But the locations are different. In the duodenum, it is coming from submucosa. In the jejunum and ileum, it is coming from mucosa. The cells which are present in mucosa are called crypts of Lieberkühn. The cells which are present in the submucosa are called Brunner's glands. Okay, now what about this, particularly about the succus entericus? So this always acts as second action. Here in this, the enzyme activator, what are present in succus entericus? What are present in the succus entericus we will see now. That is enzyme activator. This is about enterokinase. which is very much useful to activate pancreatic trypsinogen to trypsin. So what is the use of enterokinase? Enterokinase will help to convert trypsinogen to trypsin. Trypsinogen is converted into trypsin with the help of enterokinase. You see the variety. From pancreas, trypsinogen is coming. From intestine, enterokinase is coming. These two are coming from different places, but the enterokinase is useful for activating the trypsinogen to trypsin. And not only like that, not only the enzyme activator, other enzymes are there. This is tripeptidase, dipeptidase, aminopeptidase, okay, nucleotidase, nucleosidase, okay, so like that, nucleotidase, nucleosidase, okay, aminopeptidase, dipeptidase, lipase. Generally succus centricus will have almost more than the 7.5 to 7.8 pH is there. So what is this? This is all about alkaline medium. This is all about alkaline medium. And the pancreatic juice, before that we discussed, that is also almost, which is near to 8 pH. Its pH is 8. Okay, saliva is 6.8 pH. So like that, different pHs are there according to their particular places. Generally gastric juice we discussed, which is acidic, that is 1 around pH is. But what about this? This is almost all 7.5 to 7.8. Pancreatic juice is almost 8. So anyway, these are all, especially the succus centricus will work in the alkaline medium. Understood? Okay. So this is all about the particular parts of intestinal glands and intestinal juices. So in this lesson we discussed what are participating in the process of digestion. First thing is, what about the first thing? First thing is about salivary glands, the next thing is gastric glands, and next we discussed about liver, and we discussed about pancreas, and also we discussed about intestinal juice. So like that we discussed about 5 different types of glands which are participating in the digestion.